Welcome to all of you joining us. We are continuing our journey through the world of sequence alignment. In the previous segment, we talked about alignment of two sequences. However, what happens when we have many sequences, say hemoglobin from hundreds of organisms, and we want to align them into a common framework? One obvious option would be to perform pairwise alignment of each sequence with all the others. However, such a collection of pairwise alignment does not really capture the information that we need. It is like looking at an elephant from different angles, seeing each time only a partial view of the complete animal. Consider the following example for three sequences. If we compare sequence A to B, we get one picture. If we compare sequence A to C, we get a different picture. And when we compare sequence B to C, we get yet another picture. What we need is a tool that can bring all the sequences under analysis into a common framework. Such methods are known as multiple sequence alignment, MSA for short. Note that we are talking only about global multiple sequence alignment, aligning the sequences along their entire length. Local multiple sequence alignment involves looking for common motifs in multiple sequences, which is a separate challenge that requires different computational tools. For global MSA, we start by writing the sequences one below the other, and then want to insert gaps, which we designate by hyphens, at certain positions within the sequences, such that more columns will be better aligned with identical or similar nucleotides or amino acids. The intuitive goal is to achieve maximal similarity within columns while introducing the minimal number of gaps. You may remember that DNA, RNA, and proteins are continuous linear molecules. So what is the physical meaning of introducing gaps? Surely we don't mean to chop the molecules into pieces. We should remember that these molecules are three-dimensional objects, and by aligning their sequences, we actually align their structures. In fact, the utility of multiple sequence alignment was validated by showing that when structures are available for all organisms and sequence alignment is performed in parallel to structural alignment, we get similar results. However, as you know, a lot fewer structures than sequences are available in existing databases, so in most cases we rely on multiple sequence alignment to help us predict structural similarity. So what is the structural meaning of opening gaps in a sequence? Let's look at the following example. All the molecules remain continuous, but when we have gaps in the alignment, it usually means that the amino acids that exist in the regions where the other sequences contain gaps actually bulge and create loops or other structural motifs that are not present in the gapped molecules. Several methods are available to perform MSA, though unfortunately none of them can be guaranteed to find the optimal alignment. For the pairwise alignment problem, the dynamic programming approach could guarantee an optimal solution given a set of weights. However, the MSA problem belongs to a much harder set of problems for which no efficient algorithm exists. Thus, all the different algorithms can only suggest heuristics, methods that work pretty well for most cases. Most of the MSA algorithms work in an iterative manner. They start by locating the pair of sequences that are most similar, align them to each other, and then look for a third sequence that can fit into the alignment, and then the process repeats until all sequences are aligned. This process, of course, gives considerable importance to the first sequences that are chosen as a seed of the alignment. Other algorithms are more flexible and re-examine the emerging MSA to see if the process reaches a dead end, whereby the initial sequence steers the alignment in such a way that the subsequent sequences cannot be fit well into the alignment. In such cases, these algorithms backtrack and start the alignment process using different seed sequences. Let me describe one of the most popular algorithms in a little more detail. We will look at the Clustal family of algorithms, of which Clustal Omega is the latest version. Let us take as an example an MSA of 5 related proteins, the alpha and beta chains of hemoglobin from man and from horse, and myoglobin from whale. The process starts with computing using the pairwise edit distance algorithm, the distance between each pair of sequences, which are stored in a distance matrix. From this distance matrix, the algorithm builds a tree to guide the MSA construction. In this example, the algorithm decided, based on the matrix distances, to start by aligning the alpha chain of human and horse, then aligning the beta chains of human and horse to each other, then aligning these two alignments to each other. Alignment of alignments can be done by a variation of the standard edit distance algorithm. The final step is to align the whale myoglobin to the previous 4 sequences, which are already aligned. At the end of this process, we obtain an alignment of all 5 sequences into a common framework. Unlike pairwise alignment, we usually cannot assign a quantitative score to an alignment, and the impression is mostly visual. If we look at the output in this format, and note that other output formats are also available, perfectly conserved positions are marked with stars, highly conserved positions are marked with semicolons, and partially conserved positions are marked with a single dot. Usually, different types of amino acids, such as hydrophobic, hydrophilic, and charged, are marked with distinct colors. So columns in which there is a dominant color are usually highly conserved. In this example, we can see that the 4 hemoglobin proteins are well aligned, while the myoglobin, though fitting into the framework, is an outlier. What can we learn from MSA analysis? Let's look at one example showing an MSA of a section of the GP120 envelope protein of the HIV virus. The study aligned sequences from different patients and from different stages of the infection in these patients. Without going into great details, it is clear that the left N-terminal side is highly conserved between all proteins, while the right C-terminal side is much less conserved, and many of the sequences contain many gaps. Even from this quick look, we can understand, for example, that if we want to develop a vaccine against this envelope protein, we should target it to the sequence of the N-terminal domain. If we target the vaccine against the C-terminal domain, the variability will prevent the vaccine from recognizing the variants of many sequences. Maya and Ronen. In this segment, we spoke about aligning multiple sequences into a common frame. This task has many applications in bioinformatics. However, it does not allow us to determine the order in which the sequences were created and to uncover the evolutionary relationship between the sequences. Do you still remember why you came to me? You wanted to find patient 0 in a case of viral infection. We will discuss this question in our next segments that will be devoted to phylogenetic tree reconstruction.